Our first speaker is Mary Kennedy from Caltech, who will be telling us about SYNGAP in the system. I think Rick kind of gave her a promo this morning.
Yeah, so I'm going to give a completely different talk with some ideas that are not contradictory to what Rick had to say, but they're different. And I'm going to talk mostly today about experiments with one protein and a purified protein at that. This is a rare thing that plays an important role in the synapse, and that's SYNGAP. I wanted to start out by saying that I'm going to consider the properties that we've discovered about SYNGAP in the context of the regulatory system in which it's embedded. And so I'll go back and forth between experiments with the protein itself and thought experiments about the system and data about the biochemical regulatory system. In general, I think it's critical for our understanding of complex regulatory systems like this that we look carefully at the behavior of the individual proteins that are involved. Many of you who try to do that know that it's very difficult to fund such studies lately. That's a mistake. It's particularly a mistake, I think, in neuroscience, where many of the proteins are highly evolved, and they've become specialized for the very delicate regulation that is necessary for thinking and for memory formation. So I hope when you see the kinds of properties that I'm talking about, you'll see what I mean. So this is a slide that some of you saw a version of earlier that just points out the importance of understanding this protein. In about 2009, led mainly by Fadi Hamdan, it was found that about 5% of patients with non-syndromic intellectual disability have heterozygous mutations of various kinds in SYNGAP, and they seem to have arisen de novo. And these are two of those first patients. So non-syndromic intellectual disability means that they have severe cognitive disability but no identifiable morphological problems. And you can see this little girl looks perfectly normal. As they collected more data, it turned out that 9% of the patients that have non-syndromic intellectual disability and also forms of autism or epilepsy have these heterozygous mutations in SYNGAP. So it's a major cause of genetic intellectual disability, and I think it's important for us to understand what about it causes it to be so common and so devastating. The mutations in SYNGAP are almost always truncations, and they cause haploinsufficiency. In other words, they're heterozygous. Occasionally, there's a point mutation that inactivates a functional domain, but most of them are deletions. So what is this protein? This is a domain diagram slightly more detailed than the one that Rick showed. It's a big protein. It's about 135 kilodaltons. It has several identifiable functional domains. It's almost entirely localized at or near postsynaptic sites in neurons. And so the most kind of obvious in the beginning functional domain that was easy to recognize by homology with other identified proteins was this RAS-GAP domain. Wittkamp and his students at the Max Planck Institute showed a few years later that the C2 domain acting with the RAS-GAP domain confers RAP GTPase activity on SYNGAP as a whole. So for those of you who weren't here earlier, RAS and RAP are small GTP binding proteins. They're active in the cytosol when they bind GTP. They slowly hydrolyze the GTP all by themselves to GDP and become inactive at that point. The active form of these proteins, in turn, activate cascades of protein kinases of different kinds. So they're ubiquitous regulators. And GAP proteins, when they bind to the active RAS GTP, or RAP GTP in this case, increase the rate of hydrolysis of GTP. So they accelerate the rate of inactivation. So RAS and RAP and other GTP binding proteins are regulated in a push-pull fashion. The formation of the active form is regulated, and its rate of decay is is also regulated. So, so their level of activity is very carefully controlled in almost all cells, and the synapse is certainly no exception. So the carboxyl half of the protein I've labeled here as a disordered domain. All that means is that it's not predicted to form alpha or beta, alpha helices or beta sheets. It turns out to be fairly common in large enzymes like this that have a disordered domain, that this domain is a major regulator of the enzymatic activity. And this is no exception with SYNGAP. As you'll see, it's busy as a bee during synaptic activity, altering things about the activity and location of this protein. At the carboxyl end is about a 5 or 7 or so residue, depending on who you believe or who you want to listen to, site that binds to the 3 PDZ domains of PSD95, the major scaffold that we just heard about in the previous talk. Okay, so, oh yes. The early studies on the activity of SYNGAP were, many of them were done in protein that was either insoluble after expression in E. coli and partially solubilized, or bound in the PSD fraction. And that's because it sticks very tightly to membranes, and independently actually of the PDZ ligand. And we don't completely understand how that works. But Ward Walkup, who is the student who did many of the actual data, gathered much of the data that I'll show you, found that if he expressed in E. coli a form of SYNGAP that has the first 100 or so residues deleted, he could express lots of it in soluble form and purify it. And as you'll see, so Arthur Kornberg, who is a very famous early biochemist, once said, you don't waste clean ideas on a dirty enzyme. And that's certainly the case with SYNGAP. We have a much clearer picture of how it's regulated by phosphorylation since we've been able to purify it and study it in its pure form. So SYNGAP is mostly studied in mice, and mice have phenotypes that are similar to the defect in humans. Knockout mice have been made in 3 different labs, actually, in Seth Grant's lab, in Rick Hugener's lab, and in our lab. And in all 3 cases, the homozygous knockout is lethal a few days after birth, whereas the heterozygous knockout has several severe prominent phenotypes. It has behavioral learning defects and deranged synaptic plasticity that was shown by Komiyama in the mouse made by Seth Grant, and this was shown in Tom Odell's lab at UCLA. In neuronal cultures, it undergoes precocious maturation of synapses, something that might be interesting to you. And it has the neurons that are missing SYNGAP have an excess of AMPA receptors, and this was shown by Luis Vasquez in my lab and also by Kim and Hugener. It also causes the heterozygote in the grown-up animal, causes an early end to the critical period during cortical development, and that was shown by Gavin Rumbaugh and his student Clement. So this long list, actually, of phenotypes in the heterozygote is referred to by geneticists as a gene dosage effect. A gene dosage effect is actually relatively rare. In many cases, decreasing the amount of a protein by a factor of 2 doesn't cause much of a phenotype in an animal. If it did, most of us probably would be in bad shape. When it does, it usually means that that protein plays has a rate-limiting step in some critical process that is defective in the heterozygote. And so I'm going to come back to why this might be with SYNGAP later on. Okay, so what is the system that it's embedded in? This is a cartoon of some of the regulatory systems that are relevant in the postsynaptic site. Much has been written about these. I'm not going to go over them in detail. This is just to give you sort of a visual overview. As you all know, when calcium comes through at the NMDA receptor, it binds to calmodulin and a few other proteins, and it activates CAM kinase 2 and about 5 or 6 other proteins. CAM kinase 2 then phosphorylates SYNGAP, and a number of other proteins. RAS and RAP and RAC are GTP binding proteins, all of which play important roles in synaptic plasticity, and downstream of them are cascades of kinases that regulate dendritic translation. They regulate exocytosis and endocytosis of receptors, and in many cases also gene expression. So for the purposes of this talk, there's calcium. For the purposes of this talk, I'm going to focus on the molecular pathways that underlie regulation of AMPA receptor insertion. I'll point out that when calcium flows into the cell, calmodulin can activate a protein called RAS-GRIF. It can activate an adenylate cyclase, which in turn activates something called EPAC, which activates RAC. RAS-GRIF activates RAS, and of course there are many other influences not directly associated with the NMDA receptor that can lead to activated RAS. But CAMK2 also phosphorylates SYNGAP, and when it does, what I'll show you is that it changes the specificity actually for RAP and RAS, and I'll talk some about the consequences of that. So Robert Malinow showed a number of years ago that overexpressing RAP protein in neurons leads to a sort of constitutive increase in endocytosis of AMPA receptors and changes the balance of endocytosis over exocytosis. And on the other hand, overexpressing active RAS leads to an increase in the balance of exocytosis over endocytosis. So one can infer from that that changing the ambient levels of active RAS or RAP may well cause a change in the either transient or long-lasting in the net rate of exocytosis or endocytosis of AMPA receptors into the plasma membrane. So I'll point out here that Daniel Choquet has shown evidence for and proposed what I think is a really nice hypothesis that suggests that trapping of AMPA, or that the increase in AMPA receptors that occurs during synaptic plasticity involves really three different processes, two of which can be regulated by activity. One is an increase in exocytosis, as I just talked about. Another process is diffusion of the receptor over the surface of the membrane, with it occasionally bamming into the postsynaptic density. And the third process that's also regulated, and regulated quite quickly after activation of NMDA receptors, as he showed, is trapping of the AMPA receptor in the postsynaptic density. So when the trapping of the receptor as it's diffusing increases, the total amount of the receptor goes up at the postsynaptic site. So that's the three-step mechanism that Choquet has proposed and that an awful lot of evidence supports. So for this talk, I'm going to focus on, or for the first half of the talk, the first characteristic of SYNGAP I'm going to show you has to do with, as I said, its specificity for RAS and RAP. So I want to first introduce you to the assay, because this slide has a lot in it once I show you all of it. The ordinate here is the rate of hydrolysis of GTP normalized per mole of RAS or RAP. And this is just RAS all by itself, slowly hydrolyzing GTP that's been pre-bound to it. And down here is RAP itself, slowly hydrolyzing. RAP is really slow on its own, slowly hydrolyzing the GTP that's been bound to it. If you add SYNGAP in saturating amounts to this reaction, you see that the rate of hydrolysis rises quite a bit, and it rises for RAS, and it also rises for RAP. So what happens if you phosphorylate it? And that is shown here. So first of all, on this side is the result of various times of incubation of SYNGAP with CAM kinase 2 under conditions under which it gets phosphorylated. And you see that by 5 or 10 minutes, you have some increase in the rate of inactivation of RAS with these purified components, but not really much. May well have an effect, but it's not a lot. On the other hand, you see a very big increase in the rate of hydrolysis of RAP after even 1 minute of phosphorylation by CAM kinase 2. On the other hand, Lori Washburn in my lab found that CDK5 can also phosphorylate SYNGAP. CDK5 is a protein that's expressed widely in neurons. Lee Waite-Tsai's lab studied it quite a bit. And if we phosphorylate SYNGAP with CDK5, it takes a little longer, but we see a quite large increase in the rate of inactivation of RAS after phosphorylation of SYNGAP, and very little effect on RAP. So the bottom line is that phosphorylation of the protein by these two enzymes shifts the balance of inactivation activity of this protein from the sort of basal ratio of 4 to more like 2 after phosphorylation by CDK5, and more like 5 or 6 after phosphorylation by CAM kinase 2. So some of you are probably, particularly the electrophysiologists, are wondering, what does this have to do with what goes on in the neuron? And what I maintain is that very often what one finds with purified proteins is very relevant in vivo. It's really not true that it usually isn't, but of course one has to do the experiment, ask the question. And so we've done that, actually, but first I want to just walk through thought experiments about what this kind of change in specificity might do within the system that I showed you earlier. So let's say that calcium has flowed in and has activated CAM kinase 2 and also these other proteins that may be there in varying amounts in different synapses that can activate RAP and RAS. But CAM kinase 2 also phosphorylates SYNGAP and differentially increases the rate of inactivation of RAP over the rate of inactivation of RAS. And so if you follow this down, you have less activated RAP, and so you have less endocytosis, and you shift the balance toward exocytosis or addition of receptors to the membrane. Obviously, that's all other things being equal, but each of the proteins in this system have an important role to play. On the other hand, when the calcium signal kind of dies out, and if CDK5 is around, SYNGAP gets phosphorylated by CDK5, and you have an increase in the rate of inactivation of RAS compared to the rate of inactivation of RAP. And so if you follow this down, you see that the rate of exocytosis decreases and the rate of endocytosis increases. And so we suspect that in this situation, CDK5 plays a homeostatic role in the synapse, acting on SYNGAP to begin to bring the system back toward where it was before the calcium signal. So in order to ask whether these phosphorylation events actually happen in neurons and under what kinds of circumstances, we used phosphospecific antibodies to some of the sites that we'd identified on SYNGAP and did experiments in cultured neurons. And so before I show you those experiments, I'm going to show you some of the sites. We used mass spectrometry to kind of exhaustively figure out the sites phosphorylated by CAM kinase 2, and there are a lot of them. They actually aren't all shown here, but only and as again is typical of a disordered domain, there are regions where several serines can be phosphorylated, and the amount of charge builds up on that domain. But a couple of these sites are phosphorylated very quickly, and we've shown with mutants, but I won't show you, have particular effects. So for CAM kinase 2, these are 1123, serine 1123 and serine 1283. Our numbering is slightly different than Rick's, but he calls this 1138. We just use numbering for two different isoforms. The sites phosphorylated by CDK5 are serine 773 and serine 802, right here around this polyproline motif. So the sites for which we have phosphospecific antibodies are these three, and I'm going to show you what happens to them after activation of NMDA receptors. We have one for 1123 that we made in my lab and one for 765 that we made in my lab, and it turns out there's a commercial one available that recognizes this site but not this site. We don't know what effect phosphorylation of, if any, of 765 has on SYNGAP activity. But we have an antibody for it, and so we can compare its regulation to the others, and that's basically what we did. So this first graph shows, oh, I should say, so the experiment was done with primary hippocampal neurons in culture, and we simply added 25 micromolar NMDA to the neurons to get a large and synchronous activation of NMDA receptors. So this in no way is meant to mimic any physiological stimulus of any particular kind. It's meant to give a large and synchronous signal so that we can watch in bulk the downstream consequences. And so it's to be taken exactly that way and not as some reflection of what is happening during a stimulus. Although it is a reflection of the biochemical mechanisms that are brought into play by activation of NMDA receptors. And that's why we did the experiment. So site 1123, after the addition of NMDA to the cultures, shows a transient increase, and then it comes down by about 3 minutes and goes below baseline. And we know because we put in inhibitors, specific inhibitors, that this slow decrease here is caused by activation of the calcium-dependent phosphatase calcineurin. This site is regulated, you can ask me about this in the questions if there's time, but it's regulated by pathways that, by calcium, in a way that activates both a positive pathway and a negative pathway with a delay. And that kind of motif in a system is referred to as an incoherent loop. What an incoherent loop can do is cause a peak because the timing of the excitation and the inhibition are out of sync. So and that's what we see. We get this loop followed by actually a decrease below baseline of 1123. So if we do the same, actually in the same homogenate, if we look at what happened to site 773, which is phosphorylated by CDK5, we see that it falls slowly along this time period. And this decrease is caused by calcineurin because it disappears when we put in inhibitors of calcineurin. In fact, we see a very small increase, and then it just sort of comes back to baseline. So there's a little bit of activation of CDK5 by NMDA activation, but it's overwhelmed by its dephosphorylation here by calcineurin. And then site 765 of unknown consequence is certainly phosphorylated after activation of NMDA receptors, but it's not a target of calcineurin. We know from use of specific inhibitors that it begins to fall here because it's dephosphorylated by phosphatases 1 and 2, which are not strongly regulated by calcium. So the phosphorylation of these sites on SYNGAP are regulated differentially, and they're regulated in ways that we would predict or expect from the model that we've been thinking about. So they have some physiological significance. Okay, so we wondered when we got this data and thought about it whether this kind of rheostat-like regulation of the specificity of SYNGAP would account for the gene dosage effect that we saw. It could, but it's a little surprising to have a gene dosage effect for an enzymatic activity because enzymes are very powerful and you often don't need a whole lot of them to do their job. But we thought more about the characteristics of this system, and they suggest to us a possible second role for PSC95, and this role is related to data that Rick showed us and also that Aisha Dosamichi and Tom Rees got using electron microscopy, which shows that phosphorylation of SYNGAP by CAM kinase seems to cause it to move out of the postsynaptic density. So this slide is meant to sort of show a different, show the role of SYNGAP in the system from a different perspective. And it emphasizes two facts. One is that there is an awful lot of SYNGAP in the postsynaptic density. And this has been shown by our lab and by other labs and by proteomics and by practically every way you could want it to be shown. There's almost as much SYNGAP as there is PSD95. So why would that be? Another fact that it shows is that the ligand of the PDZ domain ligand of PSD95 of SYNGAP, excuse me, binds to all three of the PDZ domains of PSD95. So this is supposed to represent PSD95, and these little dots here are PDZ1, PDZ2, and PDZ3. So SYNGAP can bind to all of them. And I'll show you in a minute its affinities for the different ones. They're a little bit different, but not hugely different. This slide also shows some of the other proteins less abundant than SYNGAP, but definitely there and findable in the postsynaptic density fraction that bind to PDZ domains. And those include LRRTM proteins, that stands for leucine-rich repeat transmembrane proteins. These are proteins that this protein chaperones AMPA receptors but to the postsynaptic density by binding to the PDZ domains of PSD95, and also, as it turns out, binding to presynaptic norexin. I've also shown here neuroligin, which Tom Sudoff will talk about in his talk, which binds to PDZ3, fairly specifically of PSD95, and also binds presynaptic norexin. Both LRRTMs and neuroligin norexin can induce maturation of the presynaptic terminal when they make their interactions. And in addition, LRRTM can chaperone, and does chaperone, AMPA receptors into the synapse and binds them to it. A third protein is TARP, transmembrane AMPA receptor associated proteins, I think is what that stands for. AMPA receptor assisting proteins, I can never remember. But stargazin is one example. And TARPs are subunits of AMPA receptors that bind it to the second PDZ domain of PSD95. David Brett and others have shown that phosphorylation of TARPs by CAM kinase 2 actually increases their affinity for this second PDZ domain of PSD95. So if we just think about this data for a minute, our guess is that if phosphorylation by CAM kinase 2 causes SYNGAP to leave, to abandon the postsynaptic density, it frees up an awful lot of these sites that can bind AMPA receptor-associated proteins. And another way of saying that is it frees up slots. So some time ago, Robert Malinow proposed that there were sort of pre-existing slots in the postsynaptic density that AMPA receptors could bind to, and there was something about the induction of LTP that led to more slots. So we wanted to look more closely at the association of SYNGAP with the PDZ domains of PSD95, and to do that, Ward Walkup in my lab used a set of PDZ domain affinity columns that he invented and synthesized. And these are Sephiros beads, beads made out of carbohydrates, that have bound to them either PDZ1, PDZ2, PDZ3, or a construct that contains all of the three PDZ domains. They're made recombinantly and then purified and then bound to the columns. And he put over those columns purified SYNGAP and then looked to see what bound. And so what you see is that SYNGAP binds with reasonable affinity, and I'll show you the exact affinities in a minute, to all three of the PDZ domains, as had been shown earlier by two hybrid experiments. But if you phosphorylate the pure SYNGAP with CAM kinase 2, the binding to all three of the PDZ domains is dramatically reduced. The binding to PDZ 1, 2, 3 is actually quite high, and that isn't because there's three PDZ domains on it, because this number over here is normalized to each individual PDZ domain. So this is an avidity effect, much like one sees with antibodies. And it happens when you have more than one binding site on a single protein. It's effectively a local high concentration. But even with this very high avidity, phosphorylation by CAMK2 reduces the affinity for this piece. So Ward went on to look at to measure the affinities of SYNGAP for these different PDZ domains using Picower technology. Some of you will know what that is. If you don't, I won't explain it because it'd take me forever. And you wouldn't want to hear it anyway. But it's a way of getting actual numbers for the dissociation constants between, in this case, between SYNGAP and each of these individual PDZ domains and also these constructs that contain more than one PDZ domain. The most important, there are two actually important take-home messages from this. One is that the binding affinities for the tail of SYNGAP for PSD95 are in the nanomolar range, very tight, and in the range of affinity of things like neuroligand for PDZ domains, which was measured in the last century by Tom Sudoff, who's asleep now. He says he's not. And also that they're in the same general range as the affinity between neuroligand and neurexin. So all of these proteins are held in place or held in a meshwork by binding sites and sets of binding sites that are all within this nanomolar affinity range. So another take-home message is that even for the very high apparent affinity of SYNGAP for PDZ123, the drop in affinity after phosphorylation by CAM kinase 2 is about 8-fold. It's a big drop. And even just this site 1283 near the end of the molecule, changing it to a D, which is a phosphomimetic, drops the binding affinity for PDZ123, actually it should say 123 there, by about 3-fold. So it seems to require more than one site to get the full drop, but at any rate, I'm going to finish just by showing a cartoon of this idea that we have to sort of cement it in your brains. There are many predictions that flow from this hypothesis, many of the experiments have already been done and turn out positive. In other words, this model in which you get a rearrangement of the PSD when calcium flows in which SYNGAP takes off, and therefore slots are opened and other chaperone proteins can bind it. This model this mechanism can explain many results in the past that we didn't know how to explain before. One of which, just as an example, is that Luis Vasquez in my lab probably 11 years ago showed that neurons that are missing SYNGAP show precocious synapse formation. In other words, they form synapses about 2 days earlier than normal cells in culture. And they have a lot more AMPA receptors at the postsynaptic site. He did some rescue experiments, and he found that expressing SYNGAP, simply missing the PDZ domain ligand, couldn't rescue any of those effects. In fact, for some reason, the PDZ clusters got even bigger. So this kind of mechanism can help to explain that observation, among others. It also, to me, makes a little bit more sense than, or I shouldn't say makes more sense, because we don't think as well as evolution. But it's easier for me to imagine that this kind of an effect, or this kind of function, would cause a gene dosage effect, because the amount of SYNGAP that is present in the synapse, if it's a competitor for the binding of other proteins that need to come into the synapse in a regulated fashion, if it acts as a competitor, then its concentration will affect that competition. And it may be that if its concentration drops by 2, which I should say it does in the heterozygote, it goes down by almost exactly 2, then that would explain the gene dosage effect, and this mechanism would make sense as a gene dosage effect. So with that, I'll just reiterate that I think it's important, if we really want to understand the mechanisms of regulation at the postsynaptic site, that we look carefully at the characteristics of many of the proteins themselves, in particular the very highly evolved proteins that are specialized to do, as it turns out, I think probably more than one role at the postsynaptic site. So the contributors to this work, the main contributors are Ward Walkup, now a postdoc in my lab, and Lorraine Washburn, who was a postdoc, and these other very talented students and postdocs who have gone on to other things. And this set of both undergraduates and technicians who have contributed actually a lot to this work. We also work closely with the Caltech Proteome Exploration Lab, without which we couldn't have done these experiments. And these are the people that, that work in that lab. So I'll stop.
We have time for a few questions.
Martha.
I'm just going to ask one, and I have a lot more after.
As many as you want.
So, do you have any idea when SYNGAP appears at the synapse? Is it there? Very early
It's
animal when SAP 102?
it's synthesized. So we have looked at the rate at which it's synthesized in neurons that are developing in culture, and the timing is very similar to PSD95. It goes up as the synapses are forming. We haven't split it finely with respect to the other maggot proteins,
No,
but it comes up just right at the time synapses are forming.
I would love to know that because there's got to be important differences for having three of them.
Three.
With us. And
Yeah, there's even more than three. Right, yeah.
Yeah, but
Sure.
I'm a simplifier. But
One explanation for your results would be that could be that PDZ93 doesn't bind SYNGAP as well and interferes with its.
One of the questions.
Yes.
So whether you have looked at
No.
different binding.
No. If somebody give me money, we could do a whole bunch of experiments.
I saw some
That microphone doesn't work.
yeah,
I can't hear a word. Come down here and ask.
Is there a specific phosphatase for SYNGAP, and is it related to like scale?
Not that I know of. The phosphatases that we've looked at are calcineurin and phosphatases 1 and 2. Calcineurin doesn't have a broad specificity, but it isn't certainly specific for SYNGAP. And we haven't seen any evidence that there is a third specific phosphatase. Calcineurin and in the neuron experiment that we did, calcineurin and phosphatases 1 and 2 seem to account for the dephosphorylation.
Yes, so I was wondering for the removal of SYNGAP from the PSD, so because you principally said in your model that you would free up PDE domains, so which then could find, let's say, a slot for other membrane receptors or other things. But so there's also have been data from Bernardo's group, I think, that the LTP also removes PSD95 by chemical phosphorylation of the serine 73. So this means that both would move out. So is it time?
I haven't seen any evidence that suggests that PSD95 wholesale moves out. That in the way that SYNGAP has been shown to move out of the postsynaptic density with just a glutamate stimulation. So in all of the cases with all of this kind of regulation, the timing and the extent of the events that we've looked at needs to be measured under different conditions. If all of the PSD95 moved out of the synapse, you'd certainly get a major sort of falling apart of everything.
Just to clarify, so we didn't say that all of it comes out.
That's what I thought, yeah.
A small fraction is exchanged
Yes,
out.
which makes perfect sense.
Okay, I'll ask the last question.
Okay.
Mary, this is a super naive question because a lot of this is well beyond.
That's okay.
So,
They're good questions, always.
so this push-pull thing with SYNGAP. Rick earlier talked about the fact that there's turnover very quickly all the time,
Of-
like about 5 days or something like that.
That's
Is
of the
that-
AMPA receptors.
Of the AMPA receptors. So would that flip-flop retain the sort of seesaw balance between exocytosis and endocytosis
Yeah,
that would be
I don't
necessary
think-
to maintain
Sure.
an elevated thing through the turnover?
Sure. we haven't looked at those longer time courses. It's a great question. It's not naive at all. One thing that I will say, and the answer to that might actually reside in Edinburgh. Peter Kind has shown that, and his big group have shown that there are a number of different isoforms of SYNGAP that are spliced. There's only one gene, but they're spliced differently at the C terminus and at the N terminus. About half of the splice forms get the carboxyl terminus that has that PDZ domain ligand. The other half don't. And he's shown that there are differences. I have to go back and look at his papers to see exactly when. There are differences in the expression of those different forms under different conditions in the cell. So one thing that might happen is that when you want to, quote, consolidate a synapse, and I use the term loosely, perhaps you express more of the one that doesn't have the PDZ domain ligand, and it goes and does its job as a RAS gap, but it doesn't block the sites anymore. That's one possibility, but that all of that needs to be worked out, and I'm sure that, so I'm one of the few people in the field that never thought this was going to be simple, right? It's all of these proteins are embedded in very complex pathways that have feedback and delicate regulation, and so it's going to take us a while to sort it all out, but I think we're making great progress.